I'm going to talk about stem cells. My job is I'm head of a research unit in Pfizer, which is called Nucentis. And the work that we do on regenerative medicine, the stem cell part of the company, sits in my organisation. We also work on pain and epilepsy and many other disease areas, but my focus today is about stem cells. So this is what I'm going to cover. First of all, I'm going to take you through the basics. What type of stem cells there are, where they come from, what you can use them for and what you can't use them for. And then I'm going to talk about two different things. Stem cells as tools in the pharma industry, how we're using cells made from stem cells to improve the safety of medicines, and how we're using stem cells to help us invent new medicines that we couldn't have done before. And then in my second section, I'm going to talk about stem cells as therapeutics, where we actually use cells, products made from cells, to treat patients. And I'm going to use two examples from my own organisation. And I could have picked many examples, and there are lots of companies working in this space. But in the first example, I'm going to talk about pain and using stem cells to find new treatments for pain. And in the second area, I'm going to talk about macular degeneration, because that's one of the projects that we have ongoing. And these two sections lead very neatly into the other two presentations today on personalised medicine and on regulation. So first of all, stem cells. In overview, there are three main types of stem cells. There are embryonic stem cells, and these are the most contentious and the ones you will have heard most about. But in fact, they are the ones that are used the least in research. The second type are iPS cells, induced pluripotent cells. I'm going to go through this in a bit more detail. They are not formed from embryos, and there is much less concern about those cells, and we use them a lot in research, and increasingly so. And the third type of stem cells are the adult cells. And actually, most of the clinical trials ongoing at the moment use adult cells. And there are already several products on the market that are they're made from an adult stem cell type. And of course, adult cells have been used in therapy for 60 years. Bone marrow transplants are adult stem cells. And the basic difference between them is that the embryonic stem cell is made from the inner cell mass of a discarded embryo. iPS cells are made from a cell from an adult, usually skin, but it can be blood. And adult stem cells are still present in the body long into adulthood, and they stay in certain parts of the body, like the bone marrow and the brain and parts of the skin. And each of these cell types can be used for certain things and not for others. So let me just give you a little bit more detail on that. So first of all, how do we make embryonic stem cells? This is the detail of what people really worry about. First, you start with an egg and a sperm. Sounds like I'm talking to my kids here. And then after fusion, you have a four-stage a collection of cells, and at that point each of those cells can make everything in an embryo and actually can be transplanted and made by in vitro fertilization, can be made into another person. Once you get past that stage and you get to this stage, which is called the blastocyst, the cells there, if you take one cell, it can't make a baby, because the cells that we use are taken from here, which is the inner cell mass. And the trophoblast, the outside of that 120 cell cluster, the trophoblast is used to make the placenta and the umbilical cord and the sac. And without that, you can't make a baby. So when we make stem cell lines, they come from here. Now, the stem cell lines that come from the inner cell mass are infinitely expandable. That means one cell can divide over and over and over again. Normally, if you take a cell of any type, after 40 cell divisions, the cells die, and that's what's going on in our bodies all the time as we age. But if you take a stem cell from the inner cell mass, it will expand to fill the universe if you give it the right conditions. They will grow immeasurably, and they can also differentiate and become every single cell type in the body. And that's what is so fantastic about them. So in theory, although we can't in practice do all of this at the moment, you can make pancreatic cells to understand diabetes, you can make skin cells, liver cells to understand hepatic diseases, heart cells, blood cells. In principle, you can make every cell in the body. And that is not true of adult stem cells. So for example, if you take a bone marrow cell, you really can't make it into a neuron or a liver. You can only make it into the cells that normally come from that stem cell. You following me? Yeah, I see nods. So that is embryonic stem cells, and they were first made in 1998. So in terms of research, really relatively recently. And then things changed. Things changed in 2007 with the most amazing discovery as a scientist. We couldn't believe that you could do this. Imagine you have a cake. You inject 4 things into the cake. You come back a month later, and it's turned back into eggs and flour and sugar and butter and milk. I mean, you just wouldn't imagine that that was possible. But in essence, this is what reprogramming is. An adult cell is taken either from the skin, or now we can use blood, and it's reprogrammed. So you add 4 genes. It doesn't really matter what you call them, but these are key important genes in controlling cell division and cell expansion. So 4 genes, then you put them in a dish, culture them, and most of the cells die. 99.9% of them die. But the few that remain have been reprogrammed to look just like embryonic stem cells. And like embryonic stem cells, they will grow and divide exponentially, and they can form every cell type in the human body. And these are the 2 guys that invented them, Shinya Yamanaka and Jamie Thompson. And they'll be getting a Nobel Prize one day for this, without doubt, because it has revolutionised the sort of work that we can do in research. I did forget to say one thing on here, and that is, since the advent of making stem cells from a discarded embryo in the blastocyst stage, actually scientists have now improved and perfected a technique which can make stem cells from this stage. And the important point about making stem cells from a 4 or 8 cell aggregate is that's exactly the technology that's used in pre-implantation genetic diagnosis. So you take one cell away, but it doesn't affect the other cells. And that way, the embryo is not destroyed, but the cell that is removed can be expanded, and it's a different source of an embryonic stem cell that doesn't require the destruction of an embryo. So that's the most recent iteration of the production of embryonic stem cells. So what do we use these cells for? In essence, we actually use them at the moment for quite different things. So when it comes to embryonic stem cells, these are the gold standard. We are trying to understand how to make stem cells into particular cell types in the lab so that we can make better medicines and we can make safer medicines. So it is our reference standard. It's really what you have to include in your experiment to know that you've done the right thing and you haven't made a cell that's genetically mutated or aberrant. When it comes to induced pluripotent stem cells, the brilliant thing about this is you can make iPS cells from anybody, from me, from you, anybody in the room. All it requires is somebody that knows how to do it and some of your skin cells. And it is perfect for understanding genetic diseases. So here are just a few of the disease-specific iPS cells that you can make. And it's not just important for understanding disease, but it's important for understanding genetic differences in the cause of that disease. So, for example, for cystic fibrosis, there are more than 40 different mutations that cause cystic fibrosis. Wouldn't it be fantastic if we could make lung cells from patients with each of those genetic mutations and then test which drugs work on those cells? I'm going to give you an example of that. So we can use either cell source to make pluripotent stem cells and then differentiate them, make them into the cell type of interest. Now, actually, this is the very difficult part, because when you make a stem cell into blood or a neuron or a pancreatic cell, what you have to do is recapitulate what normally happens in development. And in development, there's a very complicated sequence of growth factors, of intrinsic factors that are released from one cell onto the adjacent cell. It's incredibly difficult to recapitulate that. But that's what you need to do to make the types of cells that we're interested in. So today, we use them for drug discovery, for disease modelling, and in a few years' time, we can think about using them for precision medicine and cell therapy. So how do we use them as tools? The first thing we use them for, and this is done pretty routinely now, and there are many companies that will provide human heart cells and human liver cells to help us with our research. Human cardiomyocytes are important because one of the major problems when we develop drugs, we have to know if they are cardiotoxic. So there are occasionally, but very dramatically, side effects from drugs that are driven by activity at heart channels. And it's hard to test for that. In the past, we've had to use dog pacemaker cells or some other method that is not a human cell. If we make human cardiomyocytes, I haven't got a video, but these are just fantastic. Has anyone ever seen them? So if you make cardiomyocytes in a dish, they beat. So all the cells join together like a functional syncytium, and that they just do what they are programmed to do, and the cells beat together like a heart beats. And you can investigate each cell and all the different components of that cell and ask what the drug would do to that cell. Does it block it, which could be a substantial liability in a drug? So this has allowed us to really understand the toxicity of some of our drugs in terms of their cardiac side effects. The same is true of liver cells. Actually, our complement of liver enzymes to detoxify the drugs that we take varies from individual to individual. Some people, you may know, are very sensitive to warfarin because they metabolize it very slowly. If you don't know that, it's extremely easy to overdose on warfarin, and you can have very serious and life-threatening consequences. So we really need to understand how our drugs are broken down, and having a battery of human liver cells allows us to do that. So in terms of making safer medicines, so far cardiomyocytes and human liver cells have been very, very helpful. Now, the Innovative Medicines Initiative has already set out on a piece of work. It's called STEM Bank. And what we're doing here, pharma companies have all come together, and we're making multiple iPS lines. And we're doing that from different types of patients, and we're also developing disease modelling methods. Now, in my area, in neuroscience, if I want to understand the cause of epilepsy, there is no way I can get a human brain cell and ask, does this drug stop the firing of that cell to make epilepsy? You just can't remove a brain cell in the way you can for cancer or you can for immunology. But now that we can make iPS cells, we revert the cells to an embryonic type, and then we can differentiate them into something that is very like a central neuron, and we can measure electrical activity of the type that would make epilepsy in patients. So disease modelling is incredibly important, and it's expensive and time-consuming. To go from a fibroblast to a neuron takes 6 months. It takes a long time to make the iPS cell, and it's very, it's technical and sophisticated work. So each company doesn't want to do this themselves. This is the ideal area for a pre-competitive consortium. We're focusing on genetic diseases where we're confident that the cause of the disorder is driven by the gene, so that when we make a cell line, we know the problem will be recapitulated in that cell. And we're starting with the nervous system, with diabetes, and with toxicology. The challenge that we've got is around how to bank and share those cells, because it would be really good to have a central repository in Europe. So this is the next piece of work that we're working on. Can we collect those cell lines? They're infinitely expandable. Why would you make more than you need when you can expand them and share them? So we can collect IPS lines from not-for-profit organisations, people with genetic mutations. Those patients are searching for cures. They're searching for people to help them develop new medicines. So we can collect cells from biotechs, pharma, academia, and the next proposal we're working on is to put them all in a central bank, industrial scale, critical that we can share them across the whole of Europe and really build partnerships with the rest of the world to understand the biology and to develop new therapeutics. Now, I'm going to skip this one because I don't have time, but let me just give you one example of the work that we're doing. So we work on pain, and there are many genetic disorders that lead to painful conditions. This one here is a set of patients that have a mutation in something called the NaV1.7 ion channel, which is responsible for carrying the nervous activity, the electrical activity from the sensing part of the neuron to the spinal cord. And if you don't have that channel, you feel no pain. Now, you might think that's a really good thing, but these are the hands of a patient who has that disorder. They're totally unaware of when they burn themselves. As a child, this person chewed off the end of their fingers. You know, kids put their fingers in their mouth. They didn't feel pain when they bit their own fingers. So really quite devastating. And also a patient that I was introduced to gave birth and broke her pelvis and was totally unaware that she had done it until her leg was pointing in the wrong direction. So this is not a nice thing to have. These patients have a mutation in the same channel, but it's hyperactive. So for them, any small signal gives them intense pain. Any small change in ambient temperature, if you raise the room temperature, they get a burning sensation. It's called Burning Man syndrome. And actually, we want to be able to do something to block those channels for patients with this type of disorder or a small fibre neuropathy, a similar disorder with an overactive mutation. So what we've been able to do is, first of all, we've been able to take embryonic stem cells and make them into sensory neurons, and that's what I'm showing you here. So you see these green fibres? They're fibres that are very similar to the fibre that runs from your finger all the way up to your spinal cord and carries the pain signal. This here is a pain signal. Actually, I'm showing you a slightly different channel, but the principle's the same. You can see the pain signal, and when a person experiences pain, you see rapid firing of the signal. And we can show in these cells that we can block that firing with some of our investigative drugs. Before we had human embryonic stem cells, or iPS cells, it was not possible to get a cell that was like a neuron and was able to fire and recapitulate the sort of activity that you see in painful neuropathic pain. So that has taken the field on a long way. And of course now we're making iPS cells from those very patients I showed you earlier. And not only can we test if our drugs that work at NaV1.7 work, but we can also test which of the existing drugs that are already out there are going to be the best for that patient. So that is precision medicine in development, and Andreas will be talking more about that. Stem cells as therapeutics. If we think about the way that we treat patients, we've started with small molecules, biologics that modify disease, but actually cell therapy allows us to correct defects that are more substantial than just a small molecule or a biologic might be able to. So we are moving towards a time of regenerative medicine and replacing defective organs and tissues. We're early on this scale, but there are some products already on the market. So I've just put an example of the cell-based products that already exist in oncology, in cartilage and bone. These are already cell-based products. None of these are made from embryonic stem cells, but they're made from adult stem cells, and most of them from expanded bone marrow cells. There are more than 100 clinical trials already underway in cell therapy. Most of them are autologous. That means I use my cells to treat me. Some of them are allogeneic, which means I expand my cells, and because I'm very generous, I could treat other people with them. So there are two different types of cell ways of using stem cells. What I would say is if you look here, there are actually very few clinical trials at the moment that use embryonic stem cells or iPS cells. And there are still a lot of challenges with iPS cells, and I think we're going to hear more about that from the next speaker. And the issue there is that we don't fully understand all of the biology yet. We know we can make cells that look like stem cells, and we can expand them and differentiate them. Are they identical? And if they're not identical, what liability does that give us in terms of therapy? So I'm going to talk about one of the programs that we have, and I'm really excited about this project because it's fascinating and it's doable and it's just really creative. And so it's a partnership we have with University College London with Peter Coffey and Lyndon de Cruz is the clinical person that we work with. And what we're attempting to achieve here is a treatment for late-stage macular degeneration. So I don't know if anybody in the room has macular degeneration or who has elderly relatives with macular degeneration, but it is a very common problem as you get into your 70s, 80s, 90s, a lot older than all of us here, but it's not uncommon. And what happens is print appears distorted. You can't see things lining up properly. If you look on a grid, you would see this sort of thing. And actually, you lose the central part of your vision, and then later, you lose all of your vision. And there are treatments for earlier stage macular degeneration, but for most people, at some point, they fail the current treatments, or they could be poorly controlled. And before they go blind, we would like to be able to treat them with a cell-based therapy. So this is the new therapy that we're creating. We've taken a human embryonic stem cell line and we've worked out how to make it into retinal pigment epithelium. And that's the cell type at the back of the eye that supports the retina, the neuronal net with which you see. And those cells are critical in taking in all the waste products of the neurons and keeping them functional and keeping them active. And people go blind with macular degeneration, not because their retina dies, but because their epithelia, the support cells, die. And what we've been able to do is to make the cells into retinal pigment epithelia, put them on a disc, and then put that disc behind the retina, and we've done some safety studies. We haven't put it into people yet, but the plan is to put it behind the fovea, the central part of the retina that's important in vision. And you could see from the earlier slide, it's the central piece that you need to protect. Now, this has a few challenges. First of all, we have to be able to control the production and differentiation of those cells. We have to know that they are 100% retinal pigment epithelial cells, that they can't cause tumours, that they're safe, we can give them to patients. We have to be able to show the quality of the product. It has to be reproducible. It has to fit all the regulatory hurdles that you need for an advanced product. And then we have to invent a device with which to inject that patch at the back of the eye. And just to show you what it looks like, this is actually a view of the patch, which is 6 millimetres by 4. It's got about 100,000 cells on it. And if you cut through it, you can see this is a retinal pigment epithelial layer. Looks very much like a normal layer. We've done safety studies in pigs. We've done safety studies in mice to show that we don't get teratomas. And we're planning to start our phase 1 studies in the UK next year. So that's just an example of the sort of things that you can do with embryonic stem cells. We couldn't use an adult cell for this. There's no stem cell from the adult that will make epithelium. You can't make it from bone marrow. You can't make it from skin. You can only make this from either embryonic stem cells or iPS cells at the moment. So that's just to give you a flavour of some of the things that we can do, the promise of stem cells. And to test that you've been paying attention, I actually have a question for you. So how many human embryos would it take to treat the world's population of late-stage macular degeneration? Let's say there are 300 million patients in the world. How many embryonic stem cells would I need, in theory, to treat the entire world's population? Yay, well done. You're absolutely right. Thank you. I will end there. I've done my job. Thank you.